Good morning, good afternoon, good evening, everyone. And my name is Henry Ndithia from the National Museums of Kenya. And I'm going to talk to you about this beautiful bird called red-capped lark. We are going to call it RCL from now. And it breeds throughout the year in equatorial tropics. And I'm going to give a perspective of Kenya. And I am going to talk to you about the role of environmental variation on nestling growth and immune function in this species. And we did this work with a number of partners, including Bakari, who was a master's student, and Dr. Kevin Matson, Dr. Muchai, and Professor Tieleman. As an introduction, growth rate and development of immune function varies widely among species and within populations. And this variation is hypothesized to reflect the adaptation of specific environmental conditions. So we find that environmental conditions probably has a lot of influence of growth rate and on development of immune function. And growth rate is associated with the pace of life. And as you may know, we have a continuum of slow to fast pace of life, with those species in the temperate north hypothesized to be fast, having a fast pace of life, while those of ours here in the tropics have a slow pace of life. And growth rate is associated with latitude. And in that case, I give an example of this study of growth rate in tree swallows in different latitudes. So we have growth rates as a function of latitude from the equator, which is 0 degrees, and which increases up to 60 degrees. And this study shows that growth rate in this study increased with a distance from the equator. And growth rate is also associated with aridity. And in this study, that is represented by this graph, where we have a growth rate on the y-axis and then aridity on the x-axis. It is a good example of how growth rate is affected by aridity. And this graph is a little bit counterintuitive because it is not straightforward. So we have to do a bit of backward thinking. So on the x-axis, we have aridity, which moves from 1.5 to 3.5. And that is the aridity of the environment. And aridity is calculated by what we call aridity index. Aridity index increases with decrease in aridity of the environment. Perhaps that is a bit complicated, but to make it simple, so aridity index in this graph increases from 3.5 to 1.5. So 1.5 on the x-axis in this case would mean this is an environment that is hot and dry, an example of a desert, and 3.5 would be an example of a cool and wet environment. So, so in this case, based on that, we therefore find that our aridity on the x-axis increases from 3.5 to 1.5. So 1.5 is very more arid and 3.5 is very wet. So our interpretation of this graph is that our growth constant K decreases with increasing aridity. So that is how that growth rate is affected by aridity. And when we come to within the tropics, a lot of studies have shown that there is large spatial and temporal variation in environmental conditions over a very small geographical space, and especially when there is difference in elevation and altitude, then you find that over a small space, of a small distance, then rainfall changes drastically, and temperature as well. And within the tropics, then we would expect that cool and wet climates would have a lot of food abundance, which is good for faster nestling growth. But then within a small spatial scale, you could have a more arid climate, which would be expected to be food deficient, and therefore the nestlings that are there leads to slow growth. And within the tropics as well, a lot of studies have also shown that rainfall is very unpredictable. And you would find that in a year, you have wet periods, and I also call them I call them periods because, for example, some of our studies show that there is not, in some species, there is no distinct season where you would call a breeding season. So in a wet environment, then you would expect that there is a lot of food abundance, leading to faster growth rate of nestlings. While during the dry period or dry season, these areas are food deficient, and therefore leading to slower growth rate of nestlings. So the question, because of this unpredictability of rainfall patterns and variation of environmental conditions within a small spatial scale, then the question that begs an answer would be how nesting growth rate evolve in relations to different climatic conditions within the tropics. And the same case with the immune function, just like the growth rate, it is immune function is associated with pace of life, with reduced investment in immune function for organisms that are known to have a faster pace of life, or the other way around. But it is also hypothesized that environmental conditions are more important factors in determining or in influencing influencing immune function, variation in immune function compared to pace of life. So when we consider pace of life and environmental conditions, then it is hypothesized that environmental conditions are more important. And because of this, the influence of environmental condition, it is expected that immune defense of organisms should match the pathogen pressure in an environment. So in environments which have a lot of pathogens, then organisms living there should be adapted in such a way that their immune function should be able to withstand the microorganisms that are within that environment. Now, and because we have defined our intratropical environment as having very large variation, then we would expect that even within the tropics, there would be a lot of variation in environmental function of adults and also of nestlings. Now, with that background, we set out with an overall objective to investigate the role of environmental conditions on growth and immune function in red-capped lark, in RCL. And Specifically, we wanted to compare nestling growth rates in and immune function in RCL in 3 locations that are climatically distinct but very close to each other, and as we are going to see in the next few slides. So SK in this case stands for South Kinangop, which is a cool and wet environment. And NK is North Kinangop, which represent a warm and wet environment, and KE it represents Kidong, which is a warm and dry environment. So we are moving from along an aridity gradient from South Kinangop to Kidong. And we predicted that areas with that are cooler and wetter, that is North Kinangop and South Kinangop, then those areas would have faster growth rate, and nestlings in those areas would have a high investment in immune function, because we would expect that these areas have high availability of food. Now, in one location only, Kidong, we also examined the consequences of parents hatching or rearing nestlings at different times of the year, and the consequences of that on the growth of nestlings and on the immune function of nestlings. So we looked at nestlings that were hatched or reared during periods with high intensity of breeding or low intensity of breeding, and also during periods with high rainfall and with low rainfall. And we also predicted here that there would be faster growth rate and more robust immune function in nestlings that are bred during high nesting intensity, because these probably would represent favorable environmental conditions that are good for breeding. And another prediction that we had is that there would be no effect of rain on nestling growth and immunity because of our results of a previous study that had seen that there is no relationship between different environmental parameters that we measured with breeding. Those included rainfall, minimum, maximum temperature, food, and nesting index. And these are our 3 study locations in blue, the topmost being warm and wet North Kinangop, followed by cool and wet South Kinangop, and the bottommost being warm and dry Kidong. And North Kinangop flooded partially. That means that there were local areas where nests were flooded. So a few of them were flooded. But in South Kinangop, there were periods of the year when the entire study location was flooded for about half a year or so, or between 4 to 6 months. And this flooding caused a lot of damage on nests and also death to nestlings. These locations, as I have mentioned, are in close proximity to each other, with North and South Kinangop being separated by only 19 kilometers. South Kinangop and Kidong are 29 kilometers apart, and North Kinangop and Kidong are 34 kilometers apart. And as I have mentioned before about orography or differences in elevation and altitude, the blue broken line represents a barrier that divides North and South Kinangop on one hand, which is which are mountain grasslands, and Kidong on the other hand, which is a lowland area, semi-arid or close to arid area. And there is a huge escarpment represented by the broken lines. And that is what influenced our belief or our assumption that there were no exchanges between these locations because of the barrier, and there were also forests that were in between these three locations, and therefore we didn't expect that birds would mix from one location to the other. And our main method was nest searching, and also biometrics measurement of body mass, wing and tarsus length of chicks that we found. And we did this simultaneously in all the three locations. And we monitored nests on day 1, which we called the hatch date, and then on day 4, day 7, and day 10. And normally nestlings would leave the nests on day 10 or between day 10 and day 12. And then we put these biometric measurements on a logistic growth curve, and and then we quantified month-to-month variation in rainfall and nesting intensity. And nesting intensity we called nest index, which is the total number of nests that we found in a month, and expressed as 10 hours of effort, so that we could standardize the effort, so that we didn't put a lot of effort in one location could not be depicted more than in others. And now I cannot move anymore. Oh yeah. Now and then after that we analyzed this mass, wing, and tarsus length in relation to nest index and rainfall in one location, that is Kidong. And now moving is and we also quantified immune function in terms of indices on day 10. So in the lifespan of the nestlings, we only took measurements on immune indices on day 10. And these immune indices includes haptoglobin, which I call HP, and then NOx, which is nitric oxide, and agglutination. And then we compared immune indices across location, and then we also looked at correlation between immune indices and nest index and rain within Kidong. And then sometimes it's difficult to move. And then we did that from 2011 to from January 2011 to March 2014. If you hear me stop talking, then it means that I cannot move my slides anymore. And then I won't touch it anymore. So, and then we measured weather parameters, rainfall, temperature, and other weather parameters that we were interested in using a local weather station in each of the locations. Now, for our results, this graph shows the results of body mass as a function of age, and and we see that our nestlings in Kidong, which is the continuous unbroken line that is not broken, had the lowest mass, but then it had the highest growth constant, which I now call it K. And
Henry, you are muted.
And now?
Now we hear you.
Yeah. And then there were no significant differences between nestlings in South Kinangop North Kinangop at hatching. Neither was there among location differences in mass on day 4, day 7, and day 10. Now for
Moala petrol, almost
for wing length,
9,000.
For wing length, which is shown on this graph as a function of age, then we also find the same kind of results like we saw on the mass, that wing length for nestlings in Kidong started low, but then the growth constant was the highest. And then nestlings in South Kinangop started with the highest wing length, but then it had the lowest K. Nestlings in South Kinangop is the lowest line on the graph, and then wing length were shorter in South Kinangop than in Kidong on day 7. So on the 7th day of the life of chicks, so we had nestlings in Kidong growing faster than those in South Kinangop, but then there were no differences in wing length between nestlings in South Kinangop and North Kinangop on day 7, or between North Kinangop and Kidong on day 7. And then again, there was no among location differences in wing length in nestlings on day 1, 4, and 10. And for tassas, we show the results there of tassas as a function of age, and nestlings in Kidong started with intermediate tassas length, but then again also with the highest K, which is the black line and the top line on among the three. And then nestlings in South Kinangop had the shortest tassas and also with the lowest K, which is the bottommost line with the small dashes. And then on day 7 and on day 1, and then on day 7, we we had nestlings in South Kinangop, which is the lower line, having a shorter tarsal compared to nestlings in in Kidong, which is the topmost line. And again, on day 1, we had nestlings in South Kinangop with shorter tarsal compared to those in North Kinangop. So again, at hatching, it means that nestlings in South Kinangop had the shortest tarsals of the 3 locations on day 1, and also on day 7 for Kidong. But then there was no among location differences in nestling tarsals in North Kinangop and Kidong for the rest of the days, day 1, day 4, day 7, and day 10. Now, for those results that I have just shown, we pulled together all the nestlings in all the 3 locations. That includes nestlings that fledged successfully and also those that did not fledge. They died because of flooding and also because of starvation and also because of predation. So these 3 groups of nestlings, they dropped along the way. And we pulled all these analysis together for the results that I have just shown you. But for the results that I'm now going to show you, we restricted this analysis only to nestlings that fledged, that were successful, that came out of the nests alive. And for these results, we see the K value, and K in this case means growth rate for different locations and for mass, and also the values that I circle in red represent K values for all nestlings pulled together, those that fledged and those that did not fledge. And the second set of the K restricted represent nestlings those that only fledged, and excluding those that died along the way. So, and for body mass in South Kinangop, the topmost line, you see that we have a K of 0.34 and K restricted of 0.43, which is an increase. And for North Kinangop, we have 0.36 for K and K restricted of 0.55. But for Kidong, the K is 0.54 and K restricted of 0.57, which is almost the same. It doesn't change much. And when we go to wing length in South Kinangop, we have 0.19 for K and 0.21 for K restricted, 0.26 for K in North Kinangop, and 0.37 for K restricted, which is an increase. But for Kidong, we have 0.39 for K and 0.39 for K restricted, which did not change. And for tarsals, for South Kinangop, we have 0.25 for K and 0.30 for K restricted, which is an increase. And for North Kinangop, we have 0.26 for K and 0.34 for K restricted, which is also an increase. But for Kidong, we have 0.37 for K and 0.36 for K restricted, which relatively remains the same. So for that, we could say that K increased for nestlings from South Kinangop and North Kinangop, the wetter and the cooler and wetter location, but it remained relatively the same for Kidong. And also K remained higher for Kidong compared to South Kinangop and North Kinangop, even after we use K restricted only, meaning that this is a confirmation that nestlings in South Kinangop and North Kinangop, these are the cooler and wetter locations, were suffering from starvation. The nestlings there were suffering from starvation, whereas in Kidong, it appears that nestlings there had enough food. Now, when we come to among location variation in immune function, now for haptoglobin, we see that Kidong had the highest haptoglobin levels, intermediate in North Kinangop and lowest in South Kinangop, but these were non-significant differences. And we also realize from this graph that there is a lot of variation within each location. Also, for agglutination, we also find the same pattern. A lot of variation within each location, but no differences among locations. And when we look at nitric oxide, the same pattern is continued, that we have a lot of within location variation in these immune indices, but no differences, not significant differences among locations, except that North Kinangop had seemed to have high values of nitric oxide. So, and for lysis, which we also measured, we only had 2 chicks out of 47 having lysis, and therefore we could not do any test among location tests for these immune indices. For Kidong, remember, we also wanted to see the consequences of parents hatching at different times of the year. When the nest index is high, and nest index in this case represents high nesting index, or if you like, high breeding intensity, when everybody in the population is breeding, and also with rainfall. So there were no differences between, there were no correlations between wing length and tassel strength with nest index or rain, but there were differences in body mass with nest index, in that at hatching, the body mass was higher when nest index was higher. So if you see where we have a red arrow, this continuous line represent body mass of body mass at hatching. So these nestlings had higher body mass when they were hatched. They had higher body mass, and on day 7, on the second graph, also represented with a continuous line showed by the red arrow, with more rainfall, 7-day-old nestlings were heavier. So we now see the influence of the time of breeding and rainfall on body mass. But then wing length, as I have said, and tarsus length were unrelated with nest index or rainfall. And all our 3 indices of immune function were also unrelated to nest index and rainfall. Now, what do these results tell us? We looked at nestlings of RCL in 3 regions that had different climates, and these regions were very close to each other. But then the growth rate of nestlings in these locations differed in ways that suggested that there was influence of female body condition and food availability for nestlings. And from our earlier study, in another study that we did in the same locations, we had measured food in these three different locations, and we found that flying invertebrates biomass was highest in Kidong, and it was very low in South Kinangop and North Kinangop. So therefore, when we see that body mass and the size of nestlings at hatching were highest in South Kinangop and North Kinangop, which are cool and wet locations, then we can presume, or we can assume, that females in these locations, because these locations have scarce food, then they invested a lot of resources in their eggs so that they laid big eggs, and so that they could give rise to nestlings that have larger body mass and larger wing length, large turtles. But then, as they grew from day 1 to day 10, there was food limitation, and therefore growth rate was limited, and therefore there was slow growth rate. But then for nestlings in Kidong, they grew faster compared to the two other locations, and therefore we could presume that parent birds in this arid location, they invested in feeding their nestlings because there was plenty of food, rather than investing a lot in female body condition and laying big eggs, and therefore that's why they had higher K. And when we further investigated whether starvation had really an effect on the growth curves of nestlings in different locations, we actually did find that growth curves, when we only calculated the growth curves for nestlings that hatched and fledged, as compared to pulling all the nestlings together, including those that died along the way, and we find that K increased in South Kinangop and in North Kinangop, but it did not increase in Kidong. And this could be interpreted to be an additional evidence that food was really limited in South Kinangop and in North Kinangop, the cool and wet locations, as compared to Kidong. Kidong did not have this food limitation. And then when we look within Kidong, in terms of the difference in growth of nestlings at different times of the year, we also find presumably the same effect of female body condition and food availability having an effect on nestling growth. So for those nestlings that were bred during the period when everybody was breeding, that is with a high nesting index, those nestlings had high body mass, meaning that probably during that period, conditions were better for breeding for females, and therefore that's why these nestlings had high body mass. And also, we found that nestlings grew better during the periods when there was more rain. And therefore, this study tells us that different parent birds in different locations use and also at different times of the year, use different strategies to be able to produce different results in terms of pace of life. So we find nestlings in Kidong growing very fast, but starting very small. Nestlings in North Kinangop and South Kinangop starting very big, but then growing very, very slowly. So these are different strategies and resulting in different paces of life. And that is within the tropics as compared to perhaps other studies that, or as compared to the northern temperate, where probably such kind of a setting would not be provided to study this kind of, or to untangle these kinds of paces of life within the tropics. So female quality and food resources are important in determining nestling growth. And remember, we saw that immune function did not vary among locations, neither did it did not vary with time within Kidong. And we thought that perhaps the reason for this is because we measured immune function only at one specific point, that is on day 10. And probably that may have complicated things. And perhaps a future study would have to look at, would have also to measure immune function at different time points, say on day 1, day 4, day 7, and day 10. And the final thing is that only 2 of our chicks had lysis out of the 47. And that would trigger our mind to think that perhaps nesting immune system is not fully developed at this very young stage. And perhaps one interesting thing that one could study is to be able to develop a trajectory of immune function development from the time of hatching up to the time of an adult bird. And that would provide a very good study, especially in a tropical setting. And that is all I can tell you for now, but this you can also read more about our work on this paper. So you can check the DOI and then look at it afterwards. So thank you very much. And perhaps if something was not clear, we can now discuss.